right, these are going to be part 3 of your notes for chapter 9. And before we get into talking about mutations, I want to go through and I want to discuss just this picture and make sure you guys can walk through all the steps. So again, your purple part is your nucleus, your blue part is your DNA, the red part is your RNA, the brown part are your ribosomes, these little like cylinder that are blue are your tRNA, and then your amino acids are your purple. So hopefully you can follow through this with me. RNA polymerase in the initiation step of transcription is going to bond to your DNA. Woo, that's fancy. It's going to bond to your DNA and separate it at the promoter region. In your second step called elongation, this RNA strand is going to be built and get longer and longer and longer as you go. Remember, it goes from the 5' end to the 3' end, and it adds on its base pairs, replacing thymine with uracil, making a complementary strand to the DNA. In the third step, it's going to be released from that ribosome, or from that DNA complex. RNA polymerase is releasing it, but it's not done yet. Between transcription and translation, it's got to go through some splicing and some other things before it gets ready. So remember, the introns get spliced out, and it also gets this poly A tail, and what you can't see right here is a 5' head cap as well. So it gets modified, and then it finally gets to leave the nucleus. Once it leaves the nucleus, it meets up with your ribosomes, which is why if you're ever looking at a picture of a cell, you've got your rough endoplasmic reticulum right outside of the nucleus, and the rough is because it's full of ribosomes, so RNA doesn't have to travel very far to make this happen. So RNA, messenger RNA, mRNA, will meet up with your ribosome, which is full of RNA and protein, in an initiation complex. In the initiation complex, for the initiation step of translation, is methionine, the tRNA that holds the methionine, your RNA, mRNA, and your ribosome. So it makes this initiation complex in step 1, and then that methionine is going to bond with another one, and then another one, and another one in the elongation step. So the tRNAs are going to come in, and the ribosome is going to catalyze this reaction and allow the amino acids to join together. That's your elongation step. And finally, the termination step is simply that it gets released and sent out to fold and then be modified in the ER and modified further in the Golgi apparatus and then do its job. Remember, there are 61 codons. Codons are the spots on the RNA. You have a set of 3 spots that will code for an anticodon on your tRNA, which will be the complement of that. Remember, U instead of T. So the tRNA will represent the coding strand, or be complementary there. Or, sorry, the template strand that we've used before. Anyway, so the tRNA is the anticodon. The codons on the RNA, they are 3 complements to each other, and then the tRNA's anticodon is what goes and gets those amino acids. But there's 61 codons, and there's only 20 amino acids. And the question is, why is the fact that there are more of one kind than the other, and only one methionine one, whereas there's 4 that code for something like, I didn't even look up, THR, or 3 that code for isoleucine? Why is that? I still want you guys puzzling that out. So finally, we're going to talk about mutations and how mutations drive evolution. Mutations can either be irrelevant, which means they don't really matter, it's not going to affect it one way or the other, or they can be, or they can affect the way that proteins are inherited. Oops, I skipped around. Some cells make mutations in copying their genetic material, or make mistakes in copying that, and then they can be irrelevant or not. So there are 2 general categories. If you look this up, they will say there are 2 general categories. One of them being germline mutations, which you can inherit with your sperm and egg, and one of them being somatic cell mutations. In your germline mutation, there was a single point mutation, which we'll talk about how this happens, a substitution mutation. In the case of Queen Elizabeth, she inherited a mutation either from her mom's egg or her dad's sperm. Remember, they both can give the X because this is found on the X chromosome. And it's a recessive trait that causes hemophilia. Well, she was just a carrier of it, didn't even know she had the mutation, but then she was able to pass on that mutation. Some of her daughters became carriers, and her son Leopold actually had hemophilia. So this is the first time doctors actually even saw this genetic disease. And it's because of one single DNA base pair substitution that happened in one of her parents that she inherited it from. So it didn't even occur in any of her other siblings as well. They could pinpoint it back to her. And then somatic can happen in your general body cells, which is what we talked about when we talked about cancer and skin cells and that kind of stuff. But for these notes, yeah, there are two general types of categories are, genetic mutations that occur when your DNA is replicating, or mutagenic mutations, which happen because of physical or chemical agents that can cause different mutations from outside the body, the environmental mutations that can happen. So either one, your cells can just make a mistake, which is a lot of like proofreading and stuff that goes into making your DNA right. So that's actually less likely than mutations that are caused initially by chemicals or any kind of other things happening to your DNA. And then when it tries to replicate as it normally would, it runs into errors. But those errors weren't caused just by your cell messing up, but they were caused by other stuff that you did to your cells. So the first kind of mutation we're going to talk about that I just briefly did when I talked about hemophilia is a substitution mutation, and it will change a single base pair by replacing one base for another. There are three kinds of substitution mutations. The first kind of substitution mutation is a silent mutation, which does not affect the sequence of the amino acid. Remember that this is kind of why you have so many tRNAs, so many codons, and so few amino acids, because you can have a substitution mistake and it can affect your DNA, but you can still end up with the same amino acid. So you've got this, for example, you've got your DNA level, which is TTC, your mRNA level, which is AAG. So the anticodon for that would be UUC, by the way, and it codes for lysine. But you can also have your silent mutation could be TTT, which is AAA, and it also codes for lysine. So if we go up to our handy little video or thing and look up lysine, which would be AA or TTT, sorry, let me find it. UUU, AAA, where's lysine? Oh, anyway, if you can find it, good on you. There it is. It's got the A's. So AAA or AAG both code for lysine. So even if there was a mutation here, it would be silent. So the polypeptide chain would turn out exactly the same, and it's all good. You don't have any problems. The second kind of mutation that we're going to talk about is a nonsense mutation. So nonsense mutations will actually result in a stop codon being where an amino acid should be, causing the translation process to stop prematurely. So you get a half-made protein, and it's just going to be degraded and used again, but this could cause major problems in any cells. So the example that it used, if the first T gets changed to an A, then its complement is UAG, and that codes for stop. And remember, there are 3 stop codons. And the last mutation that we're going to talk about is a missense mutation, and that's not necessarily where it causes it to stop, but it will change the amino acid. So if the second one gets changed to a C, that causes a missense, or if it changes to a G, that would cause a missense. And now, missense mutations aren't always bad. Sometimes they can be good. In the case of sickle cell anemia, it caused a missense mutation, so your beta globin protein got shaped incorrectly, and so it actually causes, I wish I had the picture, I forgot to get it, but it causes even a lighter color, and you have a sickle-shaped blood cell instead of the pretty biconcave disc. So again, remember, it doesn't carry oxygen as efficiently, but these guys are also semi-immune to malaria. Malaria can't attach to it because of the shape of the red blood cell is different. So it provides it a little bit of protection as long as you are heterozygous for the trait, but if you are homozygous for the trait, it can cause some major pain and major issues. So you might have a little bit of discomfort and might have a hard time breathing sometimes, or feel like it's breathing, but it's really just getting that oxygen to your body. Even though you're getting the oxygen in your body fine, it's not getting where it needs to be as efficiently as possible if you are heterozygous. But if you lived in an area like Africa, which is where this is found a lot, you have some immunities to malaria. So another type of mutation is an insertion. An insertion occurs when one or more bases are added to a DNA sequence, or a deletion occurs when one or more bases are removed from a DNA sequence. Now, anytime you have an insertion or a deletion, that can cause a frame shift mutation, and those are actually a lot more generally harmful to an organism, because it doesn't just affect that one single amino acid, it will affect every single amino acid from there on, because remember, they're done in groups of 3. So you have 3, you have 3, you have 3, but now you have 3 here. The 3 that used to be here, we added 1. So everything gets shifted down. So this is basically 1 plus 2 of that one. And then this is 1, plus 2 of this 3. You see what's happening? 1 of this 3, and then 2 of this 3, and then 1 of this 3. So it's going to change this whole code. It's going to change this code. It's going to change the next code. So every single code from there on down. And here's examples of that in actual letter form. If you deleted the 4th T here, and it will change everything from here on out. So instead of it being methionine, isoleucine, arginine, and acinine, you end up with methionine, tyronine, glycine, and then so on. So it, and notice we are 1 short at this point because these last 2 don't code for anything because it's not a full codon. So reading frame or frame shift mutations are like hard on proteins to even remotely do what they should. So common misconceptions or mutations do not always have to be drastic, or they do not always have to have negative effects. Sometimes mutations can be positive and helpful, and sometimes they can just be different, like in the case of blue eyes or red hair being mutations. And sometimes they can cause organisms to have advantages if they give you a coat collar that allows you to be more camouflage, for example. So that's transcription, that's translation, that's how they affect mutations. And in class, you did a rock pocket mouse activity where you actually pieced all this together and saw how one can change and affect the other. So I hope all that's wonderful and helpful and makes sense to you. If you have any questions, please email me or message me on Teams or whatever you need to do. I'm available. See you at some point in the future.